Hi, I'm Marcos Francisco Pérez, and I'm very excited to be joining the IBMB as a Ramon y Cajal researcher in working on epigenetics and metabolism. So despite my Spanish name, actually I'm British. I was born in the British island of Jersey, and that's where I grew up, and I did my undergraduate and master's training in the UK. And then I first came to Barcelona in 2013 to do a PhD at the Centre for Genomic Regulation, the CRG, with Professor Ben Lehner. And there, the question which I was researching was the question of what makes individuals different. And we typically think of this problem as something like nature versus nurture. But sometimes even when you hold genetics and environment constant, you still have quite a lot of differences. So the model that I used for this was a really great model that's called C. elegans. So it's a tiny round worm, but it's a very important animal in genetics. It was the first animal with its genome sequenced. It's got 3 Nobel Prizes for work done in the worm. And in this case, worms in the lab are genetically identical. They are raised in the same environment, but there's still a lot of differences. Some die younger, some have more babies, some are more stress resistant, and the question was why. So I stayed 7 years in that lab, and we found a lot of questions that we didn't know were there, and we found a lot of answers as well. And one of the main things which we found was that it's not only important the environment and the physiology of the individual in determining how they turn out, but also the environment and the physiology of previous generations. So we found out, for instance, that the age of an individual's mother makes a really big difference to how they turn out. And we found also that a mother's social environment, in terms of how many worms she perceives growing around her, according to contact and communication through chemicals called pheromones, that also makes a big difference in, for instance, the sexual maturity in the next generation. So that group was a really fantastic experience in training, but I perceived that these days, increasingly, biologists have to have a more rounded understanding of different approaches. And while I'd done only experimental work in that time, I wanted to learn how to approach biology computationally, because it seemed like being able to work with big data is really important to be able to make advances on the big questions in biology. And so to that end, I switched field, and I did a 2-year postdoc. First in the London Institute of Medical Sciences, and later at the University of Oxford with Dr. Peter Sarkis. And this was pure computational research, analysing these very large data sets which are publicly available, in which there's a wealth of information for making hypotheses and testing them too. During that time I worked on, for instance, the problem of communication between the nucleus and the organelle. And then later, something which really interested me and fit with something that I'd been interested in my worm days, which was the intersection between epigenetics and metabolism, which is what I hope to focus on here at the IBMB. The IBMB is a really fantastic interdisciplinary institute where people are trying to understand life from different perspectives and from different scales. And in the Department of Cells and Tissues, where I'm going to be working, there are people who are working and doing great research on epigenetics and also on metabolism. And so where I see myself as trying to fit in the middle ground between these two fields, I see that there's going to be a lot of people to give fantastic help and feedback, and I hope that I'll be able to create a lot of productive collaborations while I'm here. And I'm also very excited to bring that little worm, C. elegans, as a model organism to the IBMB, because although I will be using other approaches and other models, it's a very attractive organism for many questions, and it's one that no one is working with here. So I'm excited about that. My research will focus on epigenetics and metabolism and where the two things meet. So researchers typically think of epigenetics as this kind of nice and clean, almost digital process of encoding information, where you put these marks along the DNA and along the chromatin fiber with the idea of sort of bookmarking genes to tell the cell which genes to turn off and on, and so forth, and so forth. Obviously that's a huge part of it, but it's also a very physical and kind of messy process. It's a chemical process where these marks are being added, and in doing so they use metabolites, and they spit out other metabolites at the end of that process. And in that sense they're tied into the larger metabolic network of the cell. And that's kind of been recognized in previous research, where people have looked at it through the lens of sort of how this messy process of metabolism can interfere with this nice clean digital process of encoding information in these epigenetic marks. But what's not really often appreciated is the fact that there's so much DNA and so much chromatin in the cell. It's really tremendously abundant. And these epigenetic processes are then going on a massive scale, taking some metabolites and converting them into others. So in a way you could actually view epigenetic processes as metabolic processes in their own right. And that's a viewpoint that's not really very common, but it's a viewpoint which I'm hoping that we'll hope to develop and try and expand in my research. Because a lot of work is done on epigenetics. It's a tremendously well-researched topic, but it all tends to be through the same lens and the same viewpoint. And so my hope is that we can try and sort of expand that viewpoint, because some things which might not make very much sense from this kind of clean information encoding conception of epigenetics might make a lot more sense when we also can look at it from a different viewpoint, which is that of metabolism.